Sequencing proteins is another common topic that we do often in biochemistry labs. And the first step is extracting the protein of interest. I'll call it POI. And so the way we'll do this typically is if you have a bunch of cells in solution that we are interested in getting the protein from, what we'll do is we'll typically douse them or blast them with some sound waves, and this causes the cell membranes to rupture. And after we do that, we'll centrifuge the solution such that the organelles and the individual proteins will either exist in the pellet or the supernatant in the tube. And once we've done that, we can typically treat either the supernatant or the pellet with some kind of solvent to solvate the protein of interest and carry on with the protein sequencing. And also what's common is usually you'll just be given the protein already. So in that case, we can carry on to step 2. And so in step 2, now that we have the protein in its pure form and we want to know what its sequence is, what we need to do is break its disulfide bonds and block them from reforming. And so disulfide bonds are commonly formed between cysteines. And if we had some kind of amino acid primary sequence, what happens in the primary structure is we have this sequence of amino acids, but in the secondary structure, once we zoom out a little bit, if two cysteines come into contact with each other, cysteine is a unique molecule in that it contains a thiol group. So cysteine has a group that has a sulfur attached to a hydrogen. And if two cysteines are close enough, what will happen is they will spontaneously bond to each other and form a disulfide bond, which looks like this. So you'll have two sulfurs bonded to each other, and that's what this bond is here. And so the way we break the disulfide bonds is with something referred to as beta-mercaptoethanol, abbreviated BME. So BME breaks the disulfide bonds, and then we want to block the disulfide bonds from reforming because, remember, it's a spontaneous process. Block the reformation with DTT. And DTT stands for dithiol-3-etol. And what DTT does is it alkylates the thiol. And so this thiol here, if we formed some kind of sulfur carbon bond, the Gibbs free energy to form a disulfide bond would no longer be spontaneous, and so it wouldn't happen anymore. So it's essentially forming a protecting group, in essence, to keep these disulfide bonds from forming after we've hit them with BME. And so now that we have, after step 2, a long sequence of amino acids, what we do next is we're going to break the peptides with proteases. And to do that, we expose the primary sequence of amino acids to proteases such as trypsin. And trypsin is a very common one you'll find in labs. And we have several others, and trypsin is spelled with a Y, Trypsin. Trypsin, pepsin, and are two common ones. So the thing with these proteases is they have specificity. And in the case of trypsin, it has a C-terminal specificity for lysine and arginine. Sorry. And so what I mean by that, and I'll try to make this clean, lysine and arginine. So if we had some kind of segment of primary amino acid sequence, such as glycine, lysine, histidine, arginine, and glutamine, if we exposed this segment to trypsin, this is the N-terminal side, and this is the C-terminal side. The trypsin will cut after the C-terminal side of lysine and will cut after the C-terminal side of arginine. And in the case of pepsin, pepsin has a specificity for the N-terminal side of phenylalanine, abbreviated PHE, tryptophan, and tyrosine. And so to give an example as to what happens with pepsin, if we had a sequence such as valine, phenylalanine, and glycine, pepsin will come in and it will cut the N-terminal side of phenylalanine to the left if we're reading this in the the N-term to C-term direction. And so what we end up with after we have exposed our linear sequence, our linear primary sequence of amino acids to proteases such as trypsin, is we'll have a bunch of different fragments, peptides of amino acids in our solution. And so the next thing we do is we separate these peptides. And so in the case of above, we would have a glycine, lysine, a histidine, arginine, and a glutamine present in our solution. And so we want to separate these from each other. And the way we'll do that is we turn to chromatography. And in biotech labs, you'll commonly see the terms HPLC, stands for high performance liquid chromatography. And chromatography you could make a thesis on, but the gist of it is you have a stationary phase and a mobile phase, and it relies on a difference in affinity for individual unique fragments of those two phases. So to give an example, if this glycine-lysine peptide liked the stationary phase a lot more than histidine-arginine did, the histidine-arginine peptides would travel much quicker if we had some kind of mobile phase traveling through over our stationary phase. And so what you'll get out of your chromatographer would be regions in which we have very pure peptides. And so we would see a bunch of glycine-lysine peptides at one particular time interval if we had some kind of stream coming out of our chromatographer machine. And the next thing to do, now that we have identified, now that we have separated our peptides, is to perform Edmond degradation. And Edmond degradation is something that involves making our solution very basic, such that at a high pH, the N-terminal of these peptide sequences becomes very nucleophilic, or becomes nucleophilic enough to the point that the nitrogen lone pair, which would exist as NH2 connected to the alpha carbon connected to the carboxylate, as well as the R group and the hydrogen, this lone pair of electrons will perform as a very good nucleophile and attack a carbon on a molecule referred to as phenyl isocyanate. And so this carbon will form a bond with this nitrogen, and when it happens, the sequence of events that happens after that is the last amino acid on this sequence gets cut off. But so to give an example, if this R group were for that of glycine, for instance, after this N-terminal or N-terminus attacks the phenylisocyanate, what would happen after that is this bond between the carboxy carbon and the next amino acid, which would be lysine in this case, would be broken. However, what keeps Edman degradation from running through the whole molecule when it's in solution is the fact that we have to change the conditions in our solution in order to proceed to the next step. So immediately after this nucleophilic attack, this nitrogen and this carbon form an intermediate molecule that will not dissociate from the rest of the peptide until you make the solution acidic. And that's what allows us to individually cut the N-terminus amino acid from these peptide sequences, so we don't just have a bunch of N-terminals in our solution for various random amino acids. And so this concludes how we are able to sequence amino acids, proteins in biochemistry and in medicine. And I hope you guys find this useful. Let me know if you have any questions, and thanks for watching.